Hello everyone, I'm Jordan Weaver, a chemical engineering PhD candidate in the Swanson School of Engineering at the University of Pittsburgh. I'll be presenting Multicellular Spatial Model of RNA Virus Replication and Interferon Responses Reveals Factors Controlling Plaque Growth Dynamics for the Computational Modeling of Biological Systems Community Meeting of 2021. Let me start this presentation with some perspective on respiratory illnesses. Respiratory virus infections contribute significantly to death rates worldwide. Seasonal influenza viruses alone are responsible for between 290,000 and 650,000 annual deaths, and occasional, highly pathogenic pandemic strains can emerge, such as the 1918 Spanish flu, 1957 H2N2, 1968 H3N2, and 2009 H1N1 flus, which all resulted in significantly higher mortality rates. COVID-19 is possibly the defining healthcare crisis of the current generation, with tens of millions of global cases and more than 3.9 million reported deaths. Both influenza and SARS-CoV-2 are RNA viruses, with many aspects of the host's innate immune response conserved between both. In order to link molecular signaling at the site of infection and its impact on plaque growth and the occurrence of severe inflammation, we turn to computational modeling. Plaque growth assays seed the virus at a low multiplicity of infection, or MOI, and allow it to replicate across a sheet of host cells in a cell culture to form visible plaques. Our model represents a monolayer of susceptible epithelial cells, shown in blue on the right. Each cell contains an ODE model of innate immune signaling, viral replication and export, and cell death, based on a previous set of calibrated ODEs, which have been modified to include species export to an extracellular environment. We adapted an existing model of cell transitions during viral infection between susceptible, U, blue, eclipse phase, I1, yellow, virus releasing, I2, red, and dead, D, purple, cell states. The extracellular environment allows for the diffusion of virions, which lead to the formation of viral plaques, and type 1 interferons, responsible for spatiotemporal paracrine signaling. The antiviral state resulting from the interferon is an emergent property of the interactions between multiple biological scales, spatial propagation of infection and the cytokine signal in the environment, and the intracellular transduction of interferon signaling, rather than a traditional transition of cells to a distinct virally resistant state. In future work, the environment will also be home to mobile adaptive immune cells. This shows multiple plaques in a culture of cells infected with H1 influenza virus on the left, and an in silico replication of a smaller area on the right. The simulation took about 24 hours of real time to run at this scale, so while it isn't modeling an entire organ, our model is definitely capable of handling domain sizes into the visible and physiologically relevant range. We first explored the model's plaque growth dynamics. In experiments, there are two defined boundaries of the plaque, namely the outer edge of the dead cells in purple and the outer edge of the infected cells in yellow. While the increase in the number of viral particles during an infection is typically exponential, plaque growth, as measured by the change in the diameter of these two edges, is linear. A and B show experimental plaque growth assays with a linear regression line of the growth rates superimposed, while C shows our simulation of a similar assay. All three panels have different viral strains, so the growth rates are expected to differ between them. The important takeaway here is that the simulation captures this linear growth rate without any training to plaque growth data or explicit rules governing the linearity of the growth. The model does distinguish between eclipse phase, I1, and virus releasing, or I2 cells, which is not normally seen in the experimental plaque growth assays, so we have one extra line in the in silico replication. A lag phase, during which the initially transfected virus is establishing its replication and export, is seen both experimentally and in our simulations. D, E, and F show the viral titer, plaque images, and interferon concentration over time, respectively. This gives snapshots of plaque progression similar to the staining and imaging techniques performed experimentally. Finally, G, H, and I are continuous time course data for the same information. The center line is the median of 20 simulations, and the shaded area represents the 5th and 95th percentile. You can clearly see the variability in how the infection establishes itself. The viral load varies significantly during the first 20 hours, but once this infection has taken hold, the outcomes are nearly identical for all replicates. The slowest infections result in a higher interferon peak around 24 hours, but this increased cytokine response isn't enough to stop the progression of the plaque. From our previous work, we know that paracrine or cell-to-cell signaling can have a dramatic effect on the outcome of an infection. We explored this in the spatial model by increasing the efficacy of the JAK-STAT paracrine signaling pathway, as governed by STAT-P's activity parameter. To assess the impact of STAT-P activity on the dynamics, we simulated plaque growth with higher STAT-P activity over a range while recording several metrics. A shows the plaques at 80 hours post-infection for the baseline, 10 times and 100 times higher STAT-P activity. 10 times activity is enough to arrest plaque growth, and elevating further results in smaller overall plaques. B gives the progression of cell states across time for the same values. The same median, 5th, and 95th percentile norms apply here. You can still see that 10 times activity, all plaques arrest, but there's variability in when that occurs and how sharply the plaque's growth stops. C shows that a change in STAT-P activity impacts plaque growth rate, as quantified by the rate of change of the plaque at the end of the simulation. There's a mild decrease in growth rate until the critical point where arrest happens every time. You can see a few outliers arresting at lower values of STAT-P just prior to this. The area under the curve of the environmental virus and interferon, D and E, show reduced infection success and a stronger immune response for increasing values of STAT-P. Overall, this supports our earlier work's conclusions that paracrine signaling is a strong driving factor in infection outcomes, which could provide targets for future therapeutics. We also wanted to test a drug intervention's effects. Nebulized interferon alpha-2b and interferon beta are both being investigated as early clinical treatments for COVID-19. In vitro experiments also show that pre-stimulation of cell cultures with type 1 interferons is protective against SARS-CoV-1 and 2 and influenza. We simulated an infection with this pre-stimulation by exposing uninfected cells to environmental type 1 interferons, or IFN-E. After 12 hours of exposure, the IFN-E was removed before seeding a single infected cell in the center of the simulation domain. Panel A shows the plaque progression in a pre-stimulated simulation. IFN pre-stimulation clearly arrests plaque growth, while the same conditions in a field of naive cells would result in the infection and eventual death of all simulated cells. Panel B demonstrates that only the initially infected cell dies, while the proportion of eclipse phase cells steadily decreases after 20 hours, indicating a cessation of new infections. The environmental viral load, seen in C, also decreases after 20 hours. Environmental interferon in panel D indicates an elevated cytokine profile from the pre-stimulated cells versus baseline. Our interferon pretreatment results can explain the early clinical trials which show inhaled interferon beta as a promising treatment for COVID-19 through the protective effects seen after pre-stimulation. To determine how individual parameters affect the growth of the plaques, we performed local sensitivity analyses for 3 different regimes in parameter space. These were the baseline parameter set, the elevated paracrine signaling regime, and an elevated interferon diffusion regime. For each regime, we ran 20 simulations using the nominal parameter values. Then, we perturbed each parameter individually by plus and minus 25%, computed 20 simulations for each new parameter set, and performed statistical analysis on several sensitivity metrics. Statistical significance of the change in each metric from their nominal values was determined using a student's t-test. Black squares don't have a statistically significant change. The top row shows the cell state progression of a plaque growth assay under each regime to give you an idea of what regime the results are in. The baseline parameter set on the left leads to a continuous growth of the plaques. In this regime, STAT-P's dephosphorylation, the induction of IRF7, and the virus's rate of production, diffusion, and non-specific environmental clearance all have the largest effects on the outcome of the simulation. The high JAK-STAT and high interferon diffusion regimes on the middle and right have arrested plaque growth. In these regimes, the many parameters associated with the activation of paracrine signaling have the most substantial effects. What this means is that the sensitivity of all parameters change as the simulation moves between different regimes in parameter space. Clinically, we would want to move a patient towards the nearest regime that would lead to plaque arrest based on their current condition. Some treatment modalities may only be appropriate under certain conditions, or may have their efficacy reduced by a lowered sensitivity towards their intended molecular target. Additionally, these insights would have been missed by a global sensitivity analysis method like SOBAL or EFAST. Only targeted, local analyses with a careful review of the individual simulation's outcomes could really demonstrate these changes. This model demonstrates the dramatic dependence of cell culture fate on interferon. The multicellular model presented here serves as a jumping-off point for integration with immune cell action and other aspects of the body's response to infection, as the spatial cytokine information could be used for chemotaxis and adaptive immune cell responses. It is our hope to carry out these integrations collaboratively, with the goal of understanding severe SARS-CoV-2 infections and accelerating treatment development by simulating interventional strategies. Targeted experiments can bolster model predictions, and in turn inform experiments in a positive feedback loop between clinical data, experimental work, and computational effort. Finally, the methodology of creating data-calibrated, multi-scale models in CompuCell 3D based on an existing ODE or rule-based systems holds enormous potential for investigating the spatial aspects of biological systems. I'd like to thank my co-authors, Joshua Aponte Serrano, Dr. TJ Sego, Dr. James Glazier, and Dr. Jason Shoemaker. I'd also like to thank Dr. John Alcorn, Dr. Douglas S. Reed, and Dr. Catherine J. O'Malley at the Department of Immunology and the University of Pittsburgh Center for Vaccine Research for providing the plaque assay images, Emily Ackerman for her editorial help, along with the rest of the Shoemaker and Glazier labs. A preprint of the paper that this presentation was based on is available on bioRxiv. The link is shown on this slide. CompuCell 3D can be accessed at CompuCell3D.org. Regretfully, I will not be able to take your questions, but Dr. Jason Shoemaker will be available in my stead.